the esteemed management at Sree Abirami College of Pharmacy, respected faculty members, and others who are present at this event. We at GNANLex, our entire team, Dr. Nair, myself, Bessie, and others who have helped us with this presentation, would formally take this opportunity to thank you for giving us this platform to share our views on a wide-ranging, aspects of intellectual property rights. Bessie has done the right kind of introductions. She has shown you what are the different forms, what are the criteria for them to be qualified for protection through the various laws that have been enacted nationally and internationally. And Dr. Nair has covered a very wide range of subjects for you, beginning from patent grant procedures, what qualifies for a patented technology, to enforcing the patents, protecting them, valorizing them, as well as commercializing them through all the legal provisions that are available to inventors such as you. So I will now take this dialogue forward, and in accordance with what was given to me as a topic, I begin my talk on intellectual property in the context of pharmaceuticals. Are my slides visible and are they transitioning, and am I being heard clearly? If somebody can just respond, then I'll go ahead.
No, your slides are not visible.
Okay.
Your presentation has not started.
Yeah. Okay. All right.
Yeah, it's starting now.
All right.
Started.
Thank you so much.
Yes.
Okay. Thank you. And I'll just confirm transition also. Okay. So is everyone able to see my second slide?
Yes.
Okay. Thank you so much, sir. Thank you, Bessie. And okay. So I have two reasons to start with this slide. One of them being that this is a pharmacy college, and the second one being the fact that I am also a pharmacist, and it's a pleasure to interact with a room full of similarly, domain specialists. And in pharma, as we look at new drug discovery, we realize that quality, efficacy, and safety is what qualifies a molecule to be, approved for marketing by the regulatory authorities. So they have to satisfy these three primary criteria, along with, of course, a host of others. But this is what is the focus of the regulatory authorities before they approve a molecule to reach the masses. So you're looking at the triangular, three pillars there of quality, efficacy, and safety. While in patents, which is across all technological domains, it is not limited only to pharma. It is these three criteria which both Sir and Bessie have elaborated so well, that it is novelty and non-obviousness or inventive step and industrial applicability, which are the three primary criteria to qualify a technology for patent protection. So you are, you are looking at a patent law which is universal in terms of technological domains, which it, as I'm, stating it once, trade and law. Okay. Why science? Because in pharmaceuticals, there is a heavy emphasis on research. We are looking at the basic scientific principles as well as continuously breaking the barriers of science, coming up with breakthrough inventions. We are having new vaccines being evolved, new drugs being introduced every year, and moving from small chemical entities, pharmacists are today evolving large biological molecules to treat dreaded diseases like cancers and other chronic conditions. So science plays a major part in both these aspects. And then, we saw very elaborately how Bessie had told us that there are so many laws which are protecting your creations. So the Patent Act 1970 is, of course, there. There is a whole host of international arrangements and treaties to which India is also a signatory. So we as Indians can take advantage of those laws and treaties and arrangements as well to seek protection for our creations on a global basis. So, law plays a very important aspect. Law determines what qualifies for protection. Law determines how you'll enforce the protection, and law determines who is copying you and what is the reward you will get for that. And on the third part, you have trade, because if you look at the historical way in which business has evolved and how India from a very protected and very legally enforced market opened itself up in the late 90s, and then we went ahead and became a member of the World Trade Organization in 1995. My next slide will show a little more historical perspective, but just to give you a glimpse of it, we opened up our markets in 1995. We became a member of World Trade Organization. And in 2005, we had to enact product patent protection across all technological domains. And that determined how we will be trading with all other countries, how there will be an exchange of commerce, there will be an exchange of goods, there will be an exchange of services, and what will be the conditions under which we can do that. So, there is so much of an interaction and interplay of all these factors. And as pharmacists, at the end of the day, in all these factors that are impacting our practice as people who render remedies to diseases and disease conditions. Okay, what is a key element that is missing in this slide? If I can request the audience to tell me, what is the key element that is missing in this slide? What have we missed? What is our primary responsibility as pharmacists? If the admin can allow others to unmute and tell, I would be happy to hear some answers. Or if you're putting it in the chat box, then I will read it there. Anybody? Okay, so we'll not spend too much time waiting then. I think the most important aspect we are missing in all this is the patient, okay, who should be the center of all our aims and objectives, mission and vision statements. As pharmacists, our primary responsibility is towards the patient to render him free of diseases if possible, or to treat the conditions from which he is suffering. But in all this, unfortunately, the patient has gone to one end of the entire spectrum. Okay. He's nowhere there. He's probably missing in this entire ecosystem. However, there are certain things that our government has taken steps very actively, very proactively in many instances to protect the patient's interest. And as pharmacists, it is our duty to take that forward. And the Patent Act and its historical perspective is one of those examples where our government has taken very active involvement in protecting the interests of the patient. So when you're looking at the historical perspective, we're looking at the late 70s when the Patent Act was enacted. And at that point of time, pharmaceuticals in terms of products were not eligible for patent protection in India. Okay, we were allowing process patents to be granted, but product patents, that is new chemical entities, active pharmaceutical ingredients and pharmaceutical compositions and dosage forms were denied patent protection under the 1970 Act. And this continued till 1995 when India became a member of the World Trade Organization, and we had to implement what is called the trade-related intellectual property rights chapter of the World Trade Organization. So initially, the WTO was known as GATT, which is General Agreement on Trade and Tariff. It remorphed itself as WTO. And then TRIPS was a chapter under WTO. And if you became a member, you had to abide by the provisions in that chapter. And to comply with that, the Patent Act underwent several amendments, okay, concluding them by and large in 2005 to be TRIPS compliant. Under the TRIPS, it is mandatory that every member or the signatory nation enforce product patent and process patent protection. Okay, so any new invention which is complying or satisfying the patentability criteria in all fields of technology should be eligible for patent protection. This was the TRIPS provision, and India had to implement that. So that is why from 2005, we do grant product patents to pharmaceuticals as well. Now, what happened between 1970 and 1995? The pharmaceutical industry, because of the advantage of doing reverse engineering, okay, leveraged this advantage to such an extent that we became the pharmacy to the world. Okay, we were able to grow our industry. Okay, it multiplied manyfold, and we were, at the end of that particular era, were third in terms of volume, 14 in terms of value, and we were able to start producing and exporting drugs globally, and not only drugs, vaccines as well. And we were able to satisfy the market requirement of most of the countries globally, not only the leading Western nations, but also, we were exporters to the African, South American, and Southeast Asian countries. So that is what gave us a tag as the pharmacy to the world. And one of the primary contributors to that was this transition. We went from 1970 to 1995, which eventually got implemented in 2005. So if you were to look at what a patent is, and if you were to look at, what is the patentability criteria and non-patentable inventions under the Indian Patent Act, then you do realize that the patent is a monopoly, right? It excludes others from manufacturing, selling, offering for sale, importing, or marketing the invention. Okay. Only the inventor can do it himself, or he can license it to somebody whom he desires to give a license, right? So anybody other than this cannot enter the market. So what happens under these circumstances is there is only one entity which is producing the goods to satisfy the market requirement. Now, this can become a bit of a challenge, okay, in very critical goods like food and pharmaceuticals. So what did the Indian government do? The Indian government foresaw some of these issues, what can happen if there is a monopolistic situation, and they decided that the Indian Patents Act, under the amendments post TRIPS, should have certain provisions which will guard our patients from such kind of monopolistic rights that they had to offer to inventors. Okay. So while there were many non-patentable inventions from Section A to Section P of Section 3 of the Patent Act, okay, there is Section 3D, okay, 3C to some extent, 3D, 3E, 3I, and 3P, which are very much relevant to the pharmaceutical industry. Okay, so I will focus the next part of my talk on these very relevant sections, because that is what impacts us as pharmacists and our practice. Okay, so Section 3D speaks about the mere discovery of a new form of a known substance when it does not result in the enhancement of the known efficacy of that substance, or the mere discovery of a new property or new use. New use is something Sir briefly spoke about. Okay. And you're also talking of mere new use of a known process. Okay. So new forms, new use, and new use of known processes. all these under Section 3D of the Indian Patent Act are not patentable. And this is a particular clause which by and large was focused towards the pharmaceutical industry. And there is a very interesting explanation to this particular clause, which is the text below in the second bullet. For the purposes of this clause, salts, esters, ethers, polymorphs, metabolites, pure forms, particle size, isomers, mixture of isomers, complexes, combinations, and other derivatives of known substance shall be considered to be the same substance, unless they differ significantly in properties with regard to efficacy. So what this clause essentially means is, okay, let's take an example. Okay, we all know of amlodipine. Amlodipine is a very well-established marketed formulation for the treatment of hypertension. And amlodipine is marketed in its salt form, right? So when Pfizer actually initially invented amlodipine, the basic compound was invented. It was assayed for its antihypertensive properties and a patent was filed for the same. Later, Pfizer came back and filed another patent for amlodipine mesylate, okay, the methane sulfonic acid salt. Okay, now to a chemist like you and me, converting a base to an acid addition salt is a normal, obvious extension of the primary invention of the active pharmaceutical ingredient. And why would we do that? We would do that because the pharmaceutical, dosage form specialist would come and tell us that the base is not soluble. Okay, so give me a more soluble salt. Okay, so then you would think of, various salts which are available to you: hydrochloride, sulfate, mesylate, bisilate, fumarate. There are so many options: organic acids, inorganic acids. You know that a salt form or sometimes even a base form, okay, is more soluble. So then making syrups or drops or injections makes it that much less challenging. Okay, you have a more soluble salt and that solves the problem of solubility. Okay, similarly, you have polymorphs, right? What are polymorphs? Polymorphs are basically different crystalline structures which are evolved by recrystallizing the active chemical using different solvents and different process conditions. Now, what happens with different crystalline forms is the basic chemical remains the same, but these polymorphs have different physical and physicochemical properties. Okay, their therapeutic properties by and large remain the same. Okay, but because of their crystalline structure and the alignment of the atoms in the crystal, they may be more stable in terms of absorbing moisture from the air, which makes it more machinable because making a tablet form would be easier with a powder that absorbs less moisture from the air. You don't have to maintain dehumidified conditions during manufacturing. So machinability is the advantage that you get. Maybe they also have better solubility. Okay. Similarly, we know of several drugs like ambroxol, bromhexine, for example, where ambroxol is a prodrug and bromhexine is a metabolite. The metabolite is created by the processes that occur in the physiological system or in the body. But bromhexine has the mucolytic activity, okay, that is desired when you have a cough. But ambroxol is the prodrug, right? Now, if ambroxol is patented, would bromhexine be an invention? No, because it's a discovery. It is something that is naturally occurring in the body. So if bromhexine is, to be protected through a patent because you found out that there is active metabolite and it has very good mucolytic properties, then it would be Section 3D, which would prevent the patenting of bromhexine under the circumstances that ambroxol is already patented, right? So these are some of the clauses which are put in terms of explanation to this particular Section 3D. Now, why is this important? Because these kind of research is a logical progress in a new drug discovery process. You reduce particle size, you identify the active chiral moiety, the R enantiomer or the S enantiomer is more active. If you have mixtures of these two enantiomers, and you want to resolve them and have only the active one so that you cut down on the side effect. Okay. Then when you have conditions like tuberculosis or an HIV/AIDS kind of situation, you would want to combine drugs so that there is less resistance found by the organism and there is better output of the therapy that you're administering to the patient. So there are various reasons you would come up with all these alternatives, but most of them are obvious to a pharmaceutical scientist. And that is one of the reasons Section 3D was implemented in the Indian Patent Act. However, in all this, if you could look at the bold part in the line 2 of Section 3D, there is a clause which says if it does not result in the enhancement of the known efficacy. That means it is not patentable only if it is not more efficacious therapeutically. If, however, you are able to identify a polymorph or a salt or an ester or having a particular particle size of a drug, which gives better therapeutic efficacy than the known form, then that particular new form is eligible for patent protection. Okay, so there is a way out, but you have to generate that kind of data, establish without any doubt that it does give better therapeutic efficacy. Now, why is this clause so important? Okay, it's because of the particular story that I'm going to tell you in the next few slides. Okay, the international opinion on this clause was this was a demand on the pharmaceutical industry beyond satisfying the basic three criteria of novelty, inventive step, and industrial applicability. They said that India is demanding much more than the rest of the world is demanding. Now, why did this situation come up? Okay, it is because of this particular patent application filed by Novartis for Gleevec. Gleevec essentially is imatinib mesylate. Imatinib mesylate was in the 1990s an excellent drug to treat chronic myeloid leukemia. Okay, when there were not much options to treat leukemia, Novartis found this particular moiety. It was invented by a scientist called Zimmerman. So the first patent filed by Zimmerman was called the Zimmerman patent, which was actually filed in 1993. Okay, that was the first patent on imatinib mesylate filed in 1993, which was granted in the US as 5521184. Okay, now this particular patent was not filed in India because in those days, up to 1995, India did not grant product patent protection. Okay. Now, once the primary active ingredient was identified by Novartis, they immediately filed the patent. Okay. And what was written in that particular patent is if it is imatinib, it should be assumed that it includes all the acid adhesion salts as well. It is as if it is deemed to include a protection for those salts also. And they also said that it could be the mesylate or the mesylate salt. Okay. Now, after this, when research continued at Novartis, they found that this particular imatinib mesylate salt is quite hygroscopic, okay, and its solubility is not very good, which is fine because this does happen, okay. You have protected the molecule, but this particular salt form is not the best salt form that you or the best polymorph that you have thought of, okay. So you continue research to overcome this particular problem. And fortunately for Novartis, they came up with a beta crystalline form of imatinib mesylate. Okay, the beta crystalline form, which was a new polymorph of imatinib mesylate, was less hygroscopic and more soluble than the mesylate salt that was initially thought of. Okay. Now, once they found this, they applied for a patent. Now they applied in India as well because India had now opened its doors for product patenting. So this patent was filed in 1998, which actually covered the beta crystalline form of imatinib mesylate. But remember, the 1993 patent, which was granted in many countries along with the United States, okay, already said that you can have various salt forms, including the mesylate. Okay, so this particular US patent, okay, was a very relevant prior art. Okay, the prior art in fact disclosed that imatinib in its mesylate form can be administered to CML patients, chronic myeloid leukemia patients. Okay, so this patent in fact questioned the patentability of the following follow-on patent of 1998. This particular prior art or this particular prior that was accorded to Novartis disclosed the mesylate salt. So the later filing did not have an invention which was new product because the mesylate salt was already disclosed in the earlier one. Now, what did Novartis say? They said, yes, we did disclose that there can be mesylate forms of imatinib. However, the current invention is the beta crystalline form, which is a new polymorph. The problem we are facing is of hygroscopicity and solubility. We overcame that problem with this invention. So what are inventions for? To overcome problems in the particular technology. So we have come up with this invention, which basically is a more stable form. Okay, so the problem of stability has been addressed by this particular beta crystalline form of imatinib mesylate. Okay, and their own technical expert, okay, in his affidavit, stated that the difference in the bioavailability of the free base, which is just imatinib, with that of this beta crystalline form of imatinib mesylate is only 30%. And also the difference in bioavailability may be due to the difference in their solubility in water. Okay, so he did accept that the beta crystalline form was much better. Can you please mute yourself, Vivo 201A? Thank you. Yeah.
Laptop is charging.
You're still audible, ma'am. Yeah, thank you. Okay, so he said there is better bioavailability. Okay, it's because there is better solubility. However, what did the Indian Patent Office state? How did they review this patent application and what were their comments? They said there is a change of form, okay, and there is therefore, inherent properties of the new form is disclosed in the patent application. But this, inherent properties which are being disclosed in the patent application does not qualify for enhancement of efficacy. What they said was solubility and hygroscopicity of a polymorph improving over a known form of the substance is expected. Okay, there are more beneficial flow properties. There are better thermodynamic stability. There is lower hygroscopicity. All this was expected and the beta crystalline form fulfilled it. Okay, but Section 3D demands enhanced therapeutic efficacy. And this is not disclosed in the Novartis patent application. Okay, the order from the court said increased bioavailability does not necessarily lead to enhancement of therapeutic efficacy. So what the court essentially did was it split the definition of bioavailability and therapeutic efficacy enhancement and said that maybe because of change in the physicochemical properties of a substance, there is better availability of the drug in the body. But does better availability of the drug automatically convert to better enhancement of therapeutic efficacy? Maybe not. Maybe yes. If it is yes, then they say, please produce that data for that, which Novartis did not possess. Because at the end of the day, the drug is still imatinib only, whether it is in the beta crystalline form or whether it is in the basic salt form, okay, or whether it is in the base form. The scaffold which produces the therapeutic effect for CML patients is basically the imatinib moiety. Okay, and there is no change in that moiety. Whether you convert it into a salt, whether you convert it into a better polymorph, there is certainly an impact on physicochemical properties. there is certainly sometimes a benefit for the patient. Maybe there is less toxicity as well. Okay, in this case, there is lower hygroscopicity. However, that does not mean that the beta crystalline form of imatinib mesylate is more efficacious therapeutically in CML patients over the known imatinib mesylate from the Zimmermann 1993 patent. So this is what the Supreme Court said in its order. Of course, this case went through a large number of steps before it came to the Supreme Court of India. Okay, Novartis first appealed the decision of the patent examiner who rejected the 1998 patent from the Chennai Patent Office to the Intellectual Property Appellate Board, and then they went to the Chennai High Court, and then eventually they challenged the Chennai High Court's rejection in the Supreme Court, also filing another challenge which said that Section 3D itself is not constitutional or is not in compliance with the TRIPS provisions. All of these were knocked out by the Supreme Court, giving this particular argument that new forms of known substances are not patentable if they do not display enhanced therapeutic efficacy. Now, if you, as I said, look at the logical process of a drug development program, then it is natural to look at better particle size, more soluble salts, more preferred enantiomer, or a more preferred positional isomer, or even a better simple salt. Okay, but does this mean that such kind of inventions are not encouraged in India? No, they are encouraged in India, but you have to show that there is enhanced therapeutic efficacy. Okay. The Supreme Court said that we have held that the subject matter, the beta crystalline form of imatinib mesylate, does not qualify the test of Section 3D of the Act. But that is not to say that Section 3D bars patent protection for all incremental inventions of chemical and pharmaceutical substances. So why incremental? Because the basic drug has already been invented and filed as a separate patent application in the first case. Okay, the 1993 patent filed and disclosed imatinib as the base molecule or the scaffold of the base molecule, which has activity against CML. So whatever inventions you do later in terms of a new soluble salt or a metabolite or a polymorph, those are incremental from the primary path breaking imatinib disclosure. So these are incremental inventions, and it's not that we don't encourage them. We do encourage them, but there has to be a benefit in terms of therapeutic efficacy. Once that is there, then the monopolistic right through a patent will be granted for such kind of inventions. Okay. Now, what was the take home and further cascading effects of this particular Supreme Court order? Okay. So, of course, it said that India also encourages secondary patents. Okay, it has to satisfy the requirement. Okay, not merely enhance bioavailability or other expected or obvious physicochemical properties, but it should also look at enhanced therapeutic efficacy. Unless that is disclosed, no patent will be granted. It was a landmark case resulting in many other countries adopting similar provisions. So what was happening is, because of restricting the monopoly on such kind of secondary inventions, okay, there were other countries of the third world order who were looking to having better trade through TRIPS and being a WTO member, but who were also impacted by this monopolistic attitude of innovator companies. So when they realized that this provision is so beneficial to their patients, so then they also implemented similar provisions. So you have Argentina, Egypt, Philippines, Ecuador, I think even Indonesia having such kind of provisions in their patent act. So India was kind of a path-breaking light, torch bearer in terms of access to, primary inventions and pharmaceuticals to their patients, but stopping secondary inventions from being, or frivolous inventions from being, patent protected. Now, this slide will actually give you an impact on why this was looked at so keenly by third world countries. Okay. Now, what happens when multiple patents are filed? Okay. So if you're looking at remdesivir, which was patented by Gilead Sciences, and this was a antiviral drug, had very good activity against, I think, the Nipah virus. And then, they had a series of patent filings, and eventually it was used for the treatment of the, SARS and CoV, or the COVID-19 virus, right? So their first patent was filed in 2009, where they disclosed the very broad structure of remdesivir and their active salts. Then in 2012 is when they identified remdesivir as the particular API which will give them the required antiviral activity, and they covered it for treating the paramoxiviridae family. Then they came up with a 2016 filing for the filoviridae virus families, and eventually specific diastereomer of remdesivir, okay, which displayed maximum activity against several viruses, was identified, and that diastereomer was protected in 2017. Expiry of the patent to 2036. So there is a 7-year gap now. And what has happened over a period of time is, for incremental inventions, Gilead Sciences had filed patent applications, and the last patent would eventually expire in 7 years. So generics, which are much cheaper and affordable to the public, would enter the market only in 2036. Similarly, bedaquiline. Bedaquiline is a molecule invented by Janssen, a division of Johnson & Johnson. And bedaquiline was an anti-tuberculosis drug, which was invented almost 50 years, after the last one was. I think rifampicin was the last drug, or maybe pyrazinamide, or one of them. But 50 years later was the first drug for bedaquiline, which was invented. And the first, a particular patent filing was done in 2023. And then the fumarate salt, okay, of bedaquiline, which is more soluble, was invented in 2007. And the patent, life was extended by 4 years. However, because many countries, including India, have a heavy tuberculosis burden, there was a need for generics to enter the market as early as possible. So several organizations opposed the fumarate filing. There was a lot of activity around this. We were also involved as a law firm in these kind of activities. And eventually, the patent office came up with a verdict that the fumarate salt does not satisfy the requirements of Section 3D. It is not an invention in view of the first filing, which is what happened with imatinib, mesylate, and Gleevec as well, and that it is disclosed in the patent filing and would therefore not be eligible for patent protection. So this caused the introduction of generic bedaquiline 4 years prior to the expiry if the fumarate patent, where the impact of Section 3D the same kind of provision in their chloroquine, which we all know during COVID times, virus, repurposed SARS and CoV. So all these second medical uses not patentable in India, because use as a patent, patenting in Europe and US. Of course, in India, if you come up with novel delivery systems, then those novel drug delivery systems of these drugs of the Patent Act, which is again because you know that there can be a vitamin B deficiency in a patient, and they need all the they need the thiamine, riboflavin, cyanocobalamin, all of them, and you're not going to expect the patient to be very compliant if they have to swallow 3 tablets. So you put all of them into one capsule, and you expect that there is better patient compliance to this particular vitamin B deficiency therapy. So if you're just going to do that, then it is certainly not patentable because it's a very obvious invention. However, the workaround in section 3E is that if you're able to show a synergy of the ingredients, then it is eligible for patent protection. Now, what do you mean by synergistic combinations? Now, what you're looking at here is so let's take this example of augmentin. Okay, you have amoxicillin and potassium clavulanic acid, right? Now, what is clavulanic acid doing? Clavulanic acid is a beta lactamase inhibitor. Okay, it inhibits that enzyme. Now, why is the inhibition of that enzyme critical? Because when you inhibit beta lactamase cannot act on amoxicillin. And amoxicillin is a beta lactam antibiotic, but it is susceptible to degradation by beta lactamase in the body. Okay, so when you have a combination of these two, then clavulanic acid will inhibit beta lactamase, making more amoxicillin available. And therefore, amoxicillin will have better blood concentrations and therefore better activity. Okay. Now, this is the theory behind synergy. If you have an effect of a particular ingredient A as X and of that of B as Y, then typically if you add X plus Y, you should get Z, right? However, when you're talking of synergy, you're looking at X plus Y being greater than Z. Under those circumstances, you call a combination synergistic, and therefore, okay, it makes it patentable. Okay, so under the Indian Patent Act, synergistic combinations, even if they are mere admixtures, can be eligible for patent protection. Now, of course, there are some examples of mere admixtures. You have Shelcal and you have multivitamin and multimineral tablets being marketed, but there are several synergistic combinations. One of the oldest, of course, is a sulfa drug with trimethoprim, where when they are both used together in a formulation, then the inhibition of the bacteria occurs at a lower minimum inhibitory concentration. Here are some other examples. So lamivudine with zidovudine having synergistic anti-HIV activity. Similarly, telmisartan with amlodipine or curcumin with gefitinib. So if you have synergistic combinations, you have won the patent battle. Okay. However, if they are mere admixtures, then you are bound to be rejected under Section 3E of the Patent Act. And then you're looking at Section 3I, which Bessie did touch upon, of course. She did give a couple of examples of root canal treatment and so on and so forth. Okay, so the key word here is the process for medicinal, surgical, curative, or prophylactic, diagnostic, therapeutic, or other treatment. Okay, so processes or methods of treatment or methods of surgery, method of prevention are all not patentable. Okay. However, if you have a product like a surgical instrument, a new surgical instrument, or a kit for the diagnosis of a particular disease, then those are products and therefore the products are patentable. So the kit for the diagnosis of dengue is patentable, but a method of diagnosis or a process of diagnosing dengue using that kit will not be patentable under Section 3I. So it is very important to understand the difference. The product is eligible, but not the process under Section 3I. And this particular provision is there in the TRIPS itself as a non-patentable invention. So it is in the Indian Patent Act is in line with the TRIPS provisions. Similarly, what 3I also says is, treatment of animals to render them free of disease. That is, to treat them either therapeutically or to give them prophylactic, interventions. Both those are not patentable. But, to increase their economic value, if you are administering certain processes or products, then even that will not be eligible for patent protection. So a method of increasing the milk production in dairy animals such as cows, buffaloes, goats, and all, not patentable under Section 3I because you're trying to increase your economic value. Moving on further, we are looking at a situation in Section 3P, which says that traditional knowledge, which is an aggregation or duplication of known properties of traditionally known component or components. So what are we looking at here? Okay, now what happens with traditional knowledge is, you all must have heard the story of, how Haldi was trying to be patented for wound healing while we knew it for so many years. Okay, we had to identify a prior art and provide it to the patent offices. And then once that particular prior art was identified, the patent office had to reject that particular patent application. So what the Indian government has done is it has prepared a database of traditional known medications and for what they have been used. For example, the cinchona bark has quinine, it has been used for antimalarial effect. Or you have ashwagandha, the bark is known to produce energy or energy enhancing or invigorating effects. So such kind of products have been included in the database. So if you again go and say that, you say that I've extracted now quinine, from the particular cinchona bark, and it has certain characteristics and that it has antimalarial activity, then it is basically, you have just reinvented the wheel. Therefore, that particular quinine as a product will not be patentable. Maybe the process of extraction is eligible for patent, but not the product itself. Okay, so if you are talking of the same components as that already recorded in the traditional medicine, and you are talking of the same therapeutic effect as that already known through traditional knowledge, then it will not be eligible for patent protection. Okay. Now, to help us understand all this, the Patent Office has released guidelines for the examination of pharmaceutical, bio, and traditional knowledge-based inventions. So if you have any doubts, you can go and read these guidelines. They have been put down with appropriate cases and examples for each clauses that they're explaining. So they are very nicely drafted and very easy to understand. No, not much legal language there. There's a lot of technicality, which is easier for people like us to understand. So this is what is about Section 3. I end the non-patentable inventions here. However, I want to touch upon other IPRs because our talk is not only about patents, but we also have to look at other IPRs. So typically, if you look at a pharmaceutical product, then product inserts, labels, and all are eligible for copyright protection. Bottle caps, inhalers, then insulin pens kind of products can be design protected. And of course, trademarks. Trademarks in pharmaceuticals is a very big business. Okay. There can be very innovative names like Dolo or Volani or Benadryl, which don't give an indication of what the active ingredient is. But it is very, common to have names like Paranorm or Parakind, where, Paranorm could be paracetamol, but Parakind would be paracetamol of Mankind Pharmaceuticals. So, that kind of names are also very common. However, it would be nice to have innovative names. And just to give you an example of the value of trademark, okay, brands possess their own values. I'm sure you understand the power of brands. Okay, a McDonald's or a Nike or an Apple are huge brands having, their own crores of values. But Dufar was a company which operated in the up to the late 90s, which actually sold its brand name Crocin to SmithKline Beecham. Okay, so Dufar sold it to SB. And I believe in those days, the brand was sold for about 60 crores. I don't have a direct reference. That is why the question mark in the slides. But you look at what SmithKline Beecham did later. Okay, they reinvented the Crocin. They used the brand name. Okay, but they came up with a double strength syrup where they have 240 milligrams per ml of paracetamol in the syrup. Then they have Crocin Advance. Then they have Crocin Pain Relief with caffeine, which is for the relief of migraine and those kind of headaches. And with this reinvention, the repositioning of the brand with all these different variants, but with the basic name Crocin still there, indicating that paracetamol is present in that particular product. Today, they're selling about 2.5 million capsules, okay, sorry, tablets or products each day. Almost that is 30 tablets per second. As we are speaking, more than 100 tablets of paracetamol are already sold. So this is how a particular brand has been leveraged so well by SmithKline. So that is the power of a brand, the power of a trademark, the power of protecting it and enforcing it. So this is just a simple slide to explain to you that IPRs need not lie only in patents. It can be in so many other forms. And that is why that inhaler um, slide was also shown by Bessie and Sergio. So moving over to the last part of my talk, okay, this is something I try and tell every academic pharmaceutical institution, particularly because I have myself, as I said, I'm a pharmacist and I've been working in IPR portfolios for quite some time now. And I have gone to several pharma colleges. Typically, you have these, divisions there. So you have chem, you have ecology, you have sutics, you have analysis, and of course you have pharmacognosy. Sometimes some of you have biotechnology also. So how would an institution like you be able to build a pharma portfolio of patenting and other IPRs? Not very difficult because you have all the strengths that the industry has in-house. I'm sure you're all pursuing research. There are higher degrees that Sree Abirami College of Pharmacy offers. Okay, along with the fact that in your entire group of institutions, you have other, medical and paramedical institutions as well. You have nursing, you have physiotherapy. So together with them, you can do the clinical aspect. You can probably come up with some devices which can help the patient and so on and so forth. So there is so much that Sree Abirami can do in terms of innovation. And I, in this slide, have simply presented how each section in your particular college can itself be contributing to a lot of innovation happening and eventually coming up with a lot of IPRs. Okay. One of the reasons I'm doing this is also the fact that today's talk is the beginning of a journey between you and us, between Sree Abirami College of Pharmacy and other institutions and GNANLex, we would basically like to forge a long-term association with you. We would want to be your partners in your innovation journey. We would want you to have an in-house IP cell where we actively advise you, mine out your intellectual properties, advise you on how to protect them, and take them forward for commercialization and licensing and so on and so forth. So valorizing, okay, so initially securing them, enforcing them, and finally valorizing them. That is the journey of your innovation cell happening. And this is just my little kind of tool that I want to provide to you as to how each of these departments can provide their inputs and you can actually build a pipeline of pharmaceutical inventions. So your chemistry department would obviously be doing a lot of process innovations, coming up with new molecules, looking at structural activity relationships, stereochemistry, polymorphism, and so on and so forth. I'm not saying that every invention is patent eligible in India, but you can look at various other opportunities, various other options, and it's not necessary that it is only a patent which has to be licensed or commercialized. It can be know-how as well. So these are just a few pointers in that direction. And then when you have a molecule that comes out of your chemistry department, which satisfies certain requirements with regard to activity, then your SOTEX department can see how there can be a stable dosage form, which can be developed. It could be even a NDDS. You could look at relief profiles. You could look at impact of particle size, work on taste and odor, come up with improved formulations. Your ecology can look at the therapeutic activity, toxicity, maybe active metabolites, look at pharmacokinetics and assays and all that. And the basic support will come from maybe cognosy for coming up with some ingredient from a natural component, or you can have analytical method validation support from the analysis department. And most critically, if they have identified a certain impurity and that impurity can be lowered through process chemistry, then that itself can be part of your IPR portfolio. So this is how your entire staff can be involved, and there can be a lot of innovations that can be coming out of your group of institutions. So I'll conclude my talk with this. And I stopped sharing screen because I did notice a lot of questions in the chat even earlier. And any other questions that have come up now, I'll certainly want to address all of them. So if there is anything else that you want to let me know, please tell me. Otherwise, I will, look at the chat and try and answer the questions. If there are any further, please unmute yourself or put them in the chat box. Okay, so there is Swati asking a question. I think it was addressed to Bessie, but I'll try and answer that. How can startups protect their IP effectively without spending too much money? Swati, if you're into the startup, then I'm sure this is very crucial for you. Not only patent, but even trademarks and design copyright kind of IPs are also very crucial for startups. And there are several schemes available for startups. The patent office itself gives you patent practitioners aid to file patents for you. There are schemes by the state governments. Okay, I'm sure Tamil Nadu also has, Maharashtra has, I know Karnataka has. Okay, I think even Delhi has. These are meant for startups for their IP expenses. And if you've already secured an IP, there are also schemes which help you to get reimbursement on your patent expenses. And once your Indian IP is filed and you think you want to go international with it, then I think BIRAC through DBT has a scheme where they pay at least 50% of the patent charges for foreign filings as well. So there are several avenues, several plans, and several schemes available. If you're incubated in a business incubator, they would know of all of these. Otherwise, you contact one of incubators like Bessie will be able to help you to connect with some of them, and they will be able to tell you about all the possible schemes that are available to you as a startup. I hope that answers your question. Please do connect with Bessie if you have further specifics that you want to have from her. Okay, then you have Brinda asking, could you elaborate on how collaboration with industry can benefit academic IP holders? Very good question, Brinda. I think at the end of the day, an academic IP or an academic researcher is more passionate about research. Okay, you love to do research, you want to follow that, and you don't want to get into the nitty gritties of commercializing the research. Okay, so you decide if research is your passion, you've invented something very nice, then whether you want to go through the journey of commercializing, where you have several challenges. You need to find out whether it is market eligible, you need to find out whether it is safe, you need to find out whether, how to position it, how to develop from product to technology, or from research or bench level research to large scale manufacturing, scale up, challenges there, post-marketing surveillance, regulatory challenges, clinical challenges. All those are what is the steps that follow basic research. So if you want to do all of that, certainly go up the startup way. But if you don't want to do it, then the best way is to license that particular innovation or chemical or whatever you have invented to an industry and let them take it ahead. Okay, because they are good at that. They do their job well. They have developed several molecules. They know the entire process. They have a well-oiled mechanism. They have the necessary infrastructure. They have the expert staff. They know what the regulations are, and they know how to work the system. So they are good at doing that. They should be the ones who would be working it towards commercialization. So this is a win-win situation. Okay, so if you are doing the development, you being an academic researcher, research is your strength, and the industry wants to leverage that. However, the steps that follow research for commercialization is their strength, and you allow them to exercise that. So this is a win-win situation. So it is good that if you do research, okay, if you're doing basic research, give it off to the industry. If you're doing applied research, ensure that it is an industrially relevant project and you gain some commercial inputs from the industry for having solved their problem. Okay, so you will, in the process, you may get some upfront payments which you can use for further research, or you may get royalty payments once the product is commercialized. So you have a continuous revenue stream coming in. These are the other advantages that you have. Okay, I hope I've answered your question, Vrinda. Feel free to post anything else that you want. Okay, all right. High informative session, particularly for pharma graduates. Thank you so much, Tirumurugan sir, for the kind comments. I hope I've been able to fulfill your requirement of this talk. I didn't understand your earlier comment on to enhance the solubility, but maybe you wrote that in reply to somebody else. Any other questions, any other queries? Whatever I could see here, I've answered. If there is anything else, I'm happy to do so. Before we
That's all, ma'am.
Yeah, okay. All right.
Thank you,
you.
ma'am, for your enriching session.